Hi friends, welcome back to my channel Only Bio, presenting up the important points from the chapter Genes, which would certainly help you to get ready for NEET, CET, KVPY, Olympiad, and this will also help the CBSE, ICSE, IGCSE both students. Friends, the terms or the points discussed here are derived from MCQs that have appeared in the various national or state level competitive exams. Questions are formed from these terms or points either in similar or modified forms. These are beneficial as they will help you to master the important topics in limited span of time, frame expected questions, do effective revision, make your own synopsis. The more number of MCQs you solve, the better you understand and the confident you get. So a daily practice of few minutes with me on my channel can surely help you to master the topic, so stay tuned. So let's present up the points from the chapter genes from 41 to 60. Point number 41. Pyrimidines have six-membered rings with nitrogen in positions 1 and 3. There are two pyrimidines in DNA and they are named as thymine and cytosine. They differ only in the structure of the side groups. Friends, on the right hand side of the screen, please zoom it. You can see the pyrimidines and the purines, their chemical arrangement very particularly. And you will also see one more, that is uracil in case of pyrimidines, which you know is present in RNA instead of thymine. In other words, DNA does not have uracil but it has thymine. RNA has uracil but not thymine. Point number 42. Purines have each a six-membered ring identical to that of pyrimidines, and to this is attached a three-membered ring at positions 4 and 5. Positions 7 and 9 in the additional ring are also nitrogen. There are two types of purines depending on the side groups, and they are called as adenine and guanine. Next, in a nucleotide, the pyrimidines get linked to the sugar molecule by their third nitrogen, while the purines do so by their ninth nitrogen. Only one of the nitrogenous bases is found in a nucleotide. The combination of a pentose sugar and a nitrogenous base is called as nucleoside. Friends, on the right hand side of the screen, you can clearly make out the difference between a nucleoside and a nucleotide. Next, a nucleoside becomes a nucleotide with the addition of a phosphate group. Next, the diameter of a deoxyribose nucleotide is 10 Armstrong unit, that is 1 nanometer, while that of DNA is 20 Armstrong units, that is 2 nanometers. Next, the amount of purines, that is adenine and guanine in a polynucleotide is equal to the amount of pyrimidines, that is thymidines and cytosines in DNA. In the DNA molecule, two long polynucleotide chains are spirally twisted around each other. This is also called helical coiling, and the DNA is often referred to as the double helix. Friends, on the right hand side of the screen, you can see a beautiful picture depicting up the Watson and Crick model of DNA. Next, the two strands of DNA run in opposite directions and hence they are said to be anti-parallel. Next, the purine and pyrimidine bases are on the inside of the helix while the phosphate and the deoxyribose units are on the outside. The alternating sugar and phosphate groups on the outside are said to form the sugar-phosphate backbone of the DNA molecule. A DNA double helix will have two such backbones. Point number 51. There are two hydrogen bonds between adenine and thymine, whereas there are three hydrogen bonds between cytosine and guanine. Next, the hydrogen bond is a weak chemical bond between a hydrogen atom and an electronegative atom like nitrogen or oxygen. Next, because of the base pairing restrictions, the two strands of DNA are complementary to each other. Next, the diameter of the DNA molecule is 2 nanometers. The space between the adjacent bases is 0.34 nanometers. The helix completes one turn in 3.4 nanometers, and this means that one complete turn will include 10 base pairs. Point number 55. Because of the helical coiling, DNA molecule is not absolutely cylindrical, but there are grooves along its outer surface. The grooves also appear to be helically coiled, as is seen very particularly in the picture that has been shown here or displayed here on the right hand side of the screen. Please zoom in and see. When the molecule is observed from the side, the two grooves of different dimensions can be clearly seen. A smaller groove exists between the two strands and a much deeper one between the two adjacent coils. These are referred to as the minor groove and the major groove, respectively. Next, the sequence of bases along the polynucleotide is not restricted in any way, and an infinite variety of combinations is possible. It is the precise sequence of bases that determines the genetic information. Point number 58. There is no theoretical limit to the length of the DNA molecule. Point number 59. There is no distinct nucleus nor chromosomes in prokaryotes. Their genetic material, which is DNA, is represented in the form of a closed circular loop. This loop is attached to the cell membrane at least at one point. The circular DNA of the prokaryotes is often referred to as chromosome, even though the structure is different from the eukaryotic chromosome. Point number 60. The prokaryotic chromosome has 80% DNA and the rest are proteins and RNA. Friends, I'm sure the series will help you. There are many more series of different chapters which have been already uploaded. So kindly see the playlist and keep studying with me. If you're new to the channel, please subscribe and keep studying. Thank you.